Hi, guys, this is Mrs. Holmer, and this is a lecture on passive versus active transport. In case you missed any of the last few days of class, this will catch you up on all of the topics. So, let's start with the difference between passive and active transport. In passive transport, a substance is moving from high concentration to low concentration, and that does not require energy. So, we call that going down the gradient. So if you wanted to see that in terms of solutions, the particles would be going from high concentration over to where they are in low concentration. And this is no energy is required. When you're going down that hill, if you're sledding or if you're rolling a ball down a hill, it requires no energy whatsoever. On the other side, we have active transport. So active transport is when a substance moves from low concentration to high concentration. So that's like taking this boulder and pushing it up the hill from low concentration to high concentration. And so this is harder and it requires the energy. And we call this going up the gradient. So in a picture, you can see where there's low concentration, we're kind of forcing that to go where it's already in high concentration. And so that requires energy. So here's passive transport. Passive transport going down the gradient, down the hill from high concentration to low concentration. No energy. It's fun, easy, it's natural. And so that's passive transport. Up the gradient, this is when you're bringing your sled back up. You have to go from low to high. It requires energy. It's not so much fun because you're using that energy. So we call this going up the gradient or against the gradient. So in each of these cases, we can identify it as either active or passive. So in this first instance, we've got it going from low to high. So anytime you go low to high, think about going up that hill. That is active, and it will require energy to go up the hill, up the gradient. In this situation, see it's higher on the outside and lower on the inside. So we're going from high to low. So that is passive, which does not require energy. In the next example, the arrow is going out of the cell. So we're going from high concentration in the cell to low concentration outside of the cell. Again, that's passive and does not require energy. And lastly, in this picture, you can tell it's lower concentration outside of the cell, and higher concentration inside. So we're going from low to high. When you go from low to high, you're going up the gradient, and that is active transport. It will require energy. So knowing where the particles are going and if they're going from high to low or low to high will tell you whether you're using active or passive transport. The other thing we've talked about in the past, just a reminder, is how we can describe the solutions, not in terms of high or low, but in terms of hypotonic, hypertonic, isotonic. And so hypo is low, hyper is high, and iso is the same. So if we're looking outside here, the solution outside the cell has more, so it is hypertonic. And then the arrow is going from high to low, so that is passive transport. In this example, the solution outside the cell seems to have less solute, these dots. There's less of them, there's more spread out. And so that would be hypotonic. Hypo is low. And since we're going from low into the cell where it's high, this would be active transport. All right, so now that we've seen the difference between active and passive transport, we're going to just focus on passive transport. So again, passive transport is going from high to low, down the gradient, no energy required. And so there are going to be three types: diffusion, facilitated diffusion, and osmosis. So diffusion is the simplest, and technically all of the passive transports can be called a type of diffusion. So in diffusion, materials are naturally moving from where they are in high concentration to where they are in low concentration. Everything wants to spread out, everything wants to be in equilibrium and in balance. So this is very natural. In terms of the cell, we're going to look at diffusion when particles move from high to low So we're talking the solute particles. So these are the things that are dissolved, the solutes that are cute, the dots. They're moving from high to low, but they're doing so straight through the cell membrane. So you can see those phospholipids right here, the heads and the tails. It's just squeezing right past to get to the other side, traveling from high to low. So what kind of particles can do this high to low right through the cell membrane, squeezing through these phospholipids? Those are going to be small and uncharged particles, mainly gases like oxygen, nitrogen, and carbon dioxide. So again, here's that image, high to low concentration directly through the membrane, those small uncharged gas particles. This is very natural. This requires no energy. That's diffusion. And this happens every time you take a breath because those gas particles, the carbon dioxide and the oxygen, diffuse from your lungs into your blood back and forth every time you take a breath. So last question I would have is, when does diffusion stop? Well, diffusion never really stops, but when it gets to be equal on either side, so we call that isotonic or equilibrium, the particles will just continue to move back and forth through the membrane, keeping them in equilibrium. So in this image, which way will the solute move through diffusion? So remember, solute moves from high to low. So here's my high concentration, here's my low concentration. It will move up. In this image, which way will the solute move through diffusion? So again, solute moves from areas of high concentration. So I've got this high concentration to low concentration. And so the solutes are going to go into the cell, high to low through diffusion. In this image, we're going to go from high concentration in the cell to low concentration outside of the cell, high to low out of the cell. And in this case, it's kind of hard to tell, but this is drawn to be somewhat equivalent, so somewhat isotonic. So in this case, they're going to go in and out of the cell because they've reached that equilibrium. All right, so that was diffusion. Diffusion is a type of passive transport. The next thing we have is facilitated diffusion. Facilitated diffusion is the same exact thing, only these particles are a little larger or they may have a charge. So I would say they're large and in charge. Now these particles can't fit through the membrane. They just can't squeeze through. So instead, they go through a protein that allows them passageway through the membrane. So those proteins that are stuck in the membrane, they help us transport things from high concentration to low concentration. So these proteins don't actually use energy. They're just like slides that we can use to slide down into the cell. So we're going from high to low still. We still require no energy, but because these particles are a little larger, we have to use a protein to facilitate or to help the diffusion. So facilitated diffusion. You look at them side by side, they're both going from high to low, from high to low. Diffusion, though, these small particles go straight through the membrane. Facilitated diffusion, we have large charged protein particles going through a protein. All right, so there are 2 out of the 3 types of passive transport. The last one is osmosis. So osmosis is the 3rd type of transport and of passive transport, and this happens when you can't move your solute, your dots, what's dissolved. So instead of moving the solutes that are kind of stuck where they are, water is going to move instead. So osmosis is the diffusion of water through a membrane. When water moves, you've done osmosis. So we're still going from high to low. We're still not requiring energy, but by moving the water, we're going to help balance things out. So you can see in this picture here, there's a lot of water on the right and not a lot on the left. So if I move some water from the right to the left, it starts to even out my solution a little bit more. So as more and more water will travel, the solutions will end up being equal in concentration. And so osmosis is when water moves to help equal everything out. Now, water does move usually through a protein called an aquaporin. And so osmosis is technically facilitated diffusion, but it's a specialized form of facilitated diffusion because water is the particle moving, not the solute. So the biggest question that we face is which way is water going to go? What's going to happen if I put this cell in this solution? Where is water going to go? And so I have two rules. The first one I like personally. I say that water always goes in the opposite direction of the solute. So in this case, I have 50% solute. So there would be a lot of dots inside the cell and 20% solute outside the cell. So what's going to happen is my solutes are going to go from high to low. So my solutes are going to move outside of this cell into the solution. The dots that are moving are going to go from the high concentration to the low concentration. So where will water go? Water always moves in the opposite direction of the solute. So water would go in. Now, if I added the amounts of water that were in either side, you'll see that water is going from where there's a lot of water to where there's a little bit of water. And so water is still going from high to low. So osmosis, when water moves, is still no energy. It's still passive transport. It's going from high to low water. So when I ask you which way is water going to go, you think about where is the solute going to go from high to low solute. Water's going to go the opposite direction. Okay, in this, we're not going to go over all these questions, but in this image, it says which way will the solute move? So solute moves from high to low, so it's going to go in, and therefore water is going to come out. Now, is water going from high to low? Yes, it's always moving from high to low because it's a passive transport. But here's the big question. Will this cell shrink or grow? Well, if you have a balloon filled with water and water is leaving it, maybe it has a hole or something, your balloon would shrink. So this cell, because it's losing water, is going to shrink. And so if I put my cell in what we call a hypertonic solution, so this solution has more solute than the cell does. So if you put the cell in a hypertonic solution, the cell will lose water and shrink. So my saying is hyper is great. It has a greater amount of solute, but the cell is going to deflate because it's going to lose water. In this situation, again, the first question I always ask myself is which way are the dots going to move? The dots are going to move high to low through diffusion, the solute. So high to low. So they're going to move out of the cell, which means water will automatically go into the cell. Now, when you put water into a cell, just like putting water into a water balloon, that cell is going to grow. So what did I do to make this cell grow? Well, I put this cell into a solution that had less solute than the cell did. So 20% had less than 50%. Now that's a hypotonic solution. So my saying is hypo is low. It has less solute. But if you put your cell in there, the cell's going to grow. Hypo is low. The cell's going to grow because the water's going to rush in. Now, the bigger the difference in these numbers, the faster that water is going to move. So if you have a huge difference between these percentages, the water is going to go very, very quickly to try to fix that problem. Now, when will water stop moving? Just like diffusion, water will stop moving when it's in equilibrium and balanced, and then water will just go in both directions. So again, to remind you, with osmosis, water moves in the opposite direction as the solute. So in this case, the solute moves left from high to low, water goes right. In this case, the solute goes right, high to low, water goes in the opposite direction. In this case, solute goes right, high to low, water moves in the opposite direction. Takes a little bit more time looking over here. The numbers 60 versus 43. So we're going to go in for the solute from high to low, and then water is going to move in the opposite direction. So to kind of wrap up this idea of the cells growing and shrinking, if you put a cell in a hypotonic solution, so outside the cell, you have less than inside the cell, through osmosis, water's going to come in as the solute goes out through diffusion. And without water coming in, the cell is going to grow. So hypo is a lower solution, the cell is going to grow. If, however, you put a cell in a hypertonic solution, so it has more, hyper is great, but the cell's going to deflate. Why? Because the water's going to rush out through osmosis as the solute rushes in through diffusion. And so your cell is going to deflate. And there's one situation we haven't talked about. What if you're in an isotonic solution, meaning it's the same on either side, inside the cell and outside the cell? Well, my saying is, iso the same as you. Cell won't change at all. So in an isotonic solution, the materials, both the solute and the water, move in both directions, but the cell doesn't gain or lose anything. It stays the same. So let's do some example problems. In this case, on the outside, the solution has less, so it is hypertonic. The dots will move from high to low, so out of the cell. The water will go in the opposite direction, so into the cell, and the cell will grow as a result. Remember, hypo is low, the cell is going to grow. In this situation, I've got the solution outside has more, so it's hypertonic, which means the solute going from high to low is going to go in, meaning the water will come out. And when water comes out, it will shrink. So hyper is great, but the cell's going to deflate. And lastly, this is isotonic, equal on both sides. So the solute will move both ways, the water will move both ways, and the cell will stay the same. Iso, the same as you, the cell will not change at all. All right, now we've looked at osmosis and how water moves through osmosis, how a cell can get bigger and get smaller, grow and shrink based on osmosis and based on the solution that you put it in. So the solution that we put body cells in, the solution that we put plant cells in is so, so important. For a healthy body, we want our cells to be in what we call a saline solution, which is slightly salty, but that will allow water to flow both ways because it is isotonic. And so saline is healthy for our body. Water travels both ways. The cell remains healthy. Now, if for some reason the cells become dehydrated, saline will become hypotonic, which means it will rush water into dehydrated cells and help them get into a healthy size. And so we want that hypotonic to the dehydrated cell to kind of make sure that the water in hypotonic, hypo is low, the cell's going to grow. So when your cell is shrinking because it's dehydrated, we need to grow it, we're going to put it in that hypotonic solution. It just happens that saline is hypotonic to the dehydrated cell. So we're going to look at how does each type of cell respond to, so each type, the body cells respond to each type of solution. So these are blood cells, they're animal cells, and you can see that if I put it in a hypertonic, it looks like it's shrinking, the water's coming out. The isotonic, the water's going both ways, so they look very healthy. And the hypotonic, notice that water's rushing in, and they're getting big, which is not good. And then notice here, this one kind of exploded, which is definitely not good. So using these pictures here, we're going to answer these questions 1 through 6 about animal cells and osmosis. All right, Number 1, the animal cell is in a hypertonic solution, so water moves out. Hyper is great. The cell's going to deflate because the water's moving out. In an isotonic solution, the water will move both ways. Iso the same as U, so we're going to go both ways and keep it equal. In a hypotonic solution, hypo is low, the cell's going to grow because water moves into the cell. In the image, you can see that lyse means to explode or to blow up. And so when you have too much water, just like you put too much water in a water balloon, it will pop. Number 5, animal cells like to be in equilibrium with their surroundings, so they're going to prefer an isotonic solution so that they're in equilibrium. And lastly, if you place an animal cell in pure distilled water, which is very, very hypotonic, would it lyse, be normal, or shrivel? Well, in a hypotonic solution, it's going to grow, and with distilled water being super hypotonic, it will probably lyse and blow up. Now, plant cells are a little different. Notice in this case, the plant cell seems to keep its shape the whole time because of the cell wall. The cell wall helps maintain its shape. And what's shrinking is the cytoplasm and the vacuole. And so we're going to look at how the plant cell responds to the 3 different types of solutions. Number 1, if you put it in a hypertonic solution, water is going to move out and it's going to shrink. If you place it in an isotonic, it's going to move both ways and stay the same. And if you place it in a hypotonic, the water is going to move into the cell. Now, notice the first one, when we shrink the cell, it's called plasmolyzed. If you were in honors, you should have seen this in lab when we shrunk the cell with salt water. The last one is called turgid. It almost looks like the cell wants to burst, but plants actually like to be super full of water. That gives them extra strength so they can stand up and they can have a little bit of added protection, plus they have all the water they need for photosynthesis. So plants like their vacuoles to be in this turgid situation, and so they prefer hypotonic solutions. Now, if a plant was placed in salt water, which is hypertonic, we're going to shrink. That's what we saw in the lab. And then last question, and kind of very important, is what feature do plants have that keeps them from bursting in a hypotonic solution? Well, when plants like this turgid feeling, but the only way they can really survive this is because they have that cell wall helping to maintain that structure of the cell. It's hard to bust through that. Animal cells don't have that, so when they get too full, they pop. Now, we saw this in the egg lab. We put an egg in a hypotonic solution, which was distilled water, and then we put it in a hypertonic solution, which was the Karo syrup. We noticed that the hypotonic, the water was had less, so hypo was low, and the egg did grow. And then we did hypertonic, that was the Karo syrup. And then hyper is great, the cell's going to deflate. So we saw that cell deflate down to basically nothing. All right, so the review of passive transport, 3 types. Diffusion, the particles go high to low. Facilitated diffusion, the particles of solute go high to low, but we're just using a protein. So those are larger charged particles. And lastly, osmosis, water is going to move through a membrane from high to low. It goes in the opposite direction of the solute. So osmosis, water moves in the opposite direction of the solute. The next topic we're going to cover is active transport. In active transport, particles move from low to high, and we're going to require energy. So in this case, it's a lot different because we're not just rolling down the hill. It's not just going with the gradient, down the gradient. It's not easy. This is hard work. We're going to force things to go from where they're in low concentration to where they're in high concentration. And so this will require energy. The first example of this is the most basic example of active transport, and this is a protein pump. where we're going to take materials that are in low concentration and literally pump them into high concentration using energy. So ATP, that's our energy for the cell. You can see I'm zapping it with energy so it can force it from low, those solute particles from low to high. This is called a protein pump. So it's using energy to pump those materials through the protein. And this is active transport, requires energy. So a lot of things can be moved through this protein pump. You will see hydrogen ions, sodium, phosphate, and even salt that would be pumped from low to high using that protein pump, using energy, which is active transport. So here it goes. We're low concentration, we put some energy in, we pump it to high concentration. How is this protein pump different from the diffusion we just talked about? Well, diffusion and facilitated diffusion go from high to low, requiring zero energy. But when you're trying to go backwards, you're going up the hill, up the gradient from low to high, we have to use that ATP energy to pump it in. And so that uses energy. That is active transport, not passive. There are two other types of active transport. One is called endocytosis. So you're going to see here the cell membrane kind of makes a pocket and brings material in, and then a vesicle comes and merges with the cell membrane to throw the materials out. This is referred to as bulk transport. It is an active transport. It requires energy, but we have two types. Endocytosis, when we make a vesicle to pull it in, endo, enter, and exocytosis, when the vesicle merges to push it out. Exocytosis, exit. So in a nutshell, we have two main types of transport: active transport that requires energy, our protein pump, and endo and exocytosis, and passive transport that does not require energy, osmosis when the water moves, diffusion through the membrane, and facilitated diffusion of larger particles through a protein in the membrane. Passive is all high to low, no energy. Active is all low to high, requires energy. That's all I have for active and passive transport and cell transport. Let me know if you have any questions, and have a good rest of your day.